I'm happy to welcome our very first speaker, which is Thomas Sauter. So he's a professor in system biology and a member of the Department of Life Science and Medicine, right? So thank you very much, Thomas, for being our first speaker. Yeah.
This one, yeah.
Yeah, this one. You have a, well...
I'll just use, yeah, okay. Pointer. No, this was not the right one. Sorry, I used the wrong button now already. Thanks. Just trying to find the pointer. The laser, it's here? Or?
No,
You can. Yeah.
But this works.
Just this one.
Yeah.
Okay. I think it's on already. It's just not working anymore. Maybe you need to restart. It's not working.
The remote control at least.
Yeah, it worked just a second ago, but maybe now it's working. Okay, perfect. Okay, thanks. Yeah, welcome also from my side. Thanks for joining today. And yeah, I'm happy to introduce a bit what we are doing at the Department of Life Sciences and Medicine. And is the sound okay like that? Yeah, good. I would like to, because this is a very general introduction, I would really use the first half of my talk to talk about life sciences and what interests me in data science. And in the second half, I will then go over two of our applications, but I'll try to do that pretty high level so that it's really more a general introduction in our work and the topics we are looking into. So let me start then. Once again, I'm trying. Okay, perfect. So I'm working in systems biology, and this is basically a summary of our daily work and my life. So not sure if this pointer is now going to work. Probably not. Not dare to touch anything here? No. Okay, so we have these three areas in systems biology which really very closely work together. So biology and then the technology for data generation and computation. And I'm considering myself as a biologist, so I'm really interested in working on diseases like cancer or Parkinson's disease and trying to help develop the treatments on these fields. But our research, I'm basically not in the lab anymore or anything like that, so I'm really working on the computer all day and basically in this transition between technology, computation, and then moving back to biology. That's my field. So biology nowadays is really driven a lot by new technologies, and these technologies allow us to generate a lot of data in biology. I'll have a few slides on that. And there is a large need of analyzing and integrating data and extracting knowledge from these data. And so that's also where my interest comes from, working together with Yannick on this master in data science, because I think that's really like the bottleneck in life sciences research. So make use of all this big data we are generating in a much more efficient way than what we are currently doing. And then, of course, on the other hand, we also need to have this transition between computation and biology. So we need to come up with new ideas and convert that back into biological language. So I usually, we had just last week, we have the teaching evaluation, and the committee asked, okay, what's your vision on for the students? And I usually answer then that that's my vision, that we need students who are bilingual. So they need to speak biology, but they also need to speak computation and math and statistics, so that they can fluently and efficiently transition between these two fields and use the methods of data science really to dig deeper in the life sciences data. Okay, so is this moving? Yeah. So we are basically in a very interesting period in life sciences. So we are in the last 20 years with all these new technologies which were developed, we basically have the capacity to move and zoom into life, so into understanding much more the molecular details of what's going on within a cell. So cell is the basic building block of life, and if you zoom in, you can find many, many things, and we have all this genome information now piling up. We know about what's happening on the epigenome, so these additional modifications which are also important for regulation. We can measure proteins and how these proteins assembly to different complexes and start to produce functionality. And then we have networks of proteins interacting, or networks of genes, transcriptional networks interacting, and they give rise to functionalities again. And I think what we try to do is, by, on one hand, dig very deep and try to, basically, it's like we are opening a window into the cell. So that's really completely fascinating to see how many details are in there. And I think there's still a long way to go to fully measure and understand that. On the other hand, we would need to also zoom out again, because in the end we are interested in what's really happening in the cancer cell, for example. So what's going wrong there, that we have the cancer growing, and where could we interfere? So which of all these details are important to understand and important to tackle? So, and it's a bit like, maybe you have kids, and then Christmas time is coming, and you have this big box of Lego pieces coming with 2,000 parts, and you see on the box, okay, this should be a car which is driving, and then you open it and you have these 2,000 pieces, and you don't have a clue how that should ever be a car which is driving. And that's a bit the situation, like we are having that. So we are seeing all these pieces, and we try to understand, okay, without having the assembly plan, try to understand how do you get the function of a cell, and what's going wrong if the function goes in the wrong direction. Okay, it's a bit sensitive. Okay, so over the last years, these omics technologies developed. And that basically allows us to quantify on a molecular level a lot what's going on. So we can measure the genome with the genomics, or we can then measure the mRNA with the transcriptomics and the protein level, proteomics, and so on. And all these different omics allow us in the end, I'm really sorry, I need to be a bit more. Allow us on each level to measure tens of thousands of entities. So each measurement of transcriptomics gives us maybe 20 to 50,000 mRNA levels. Or on the proteins, depending on the measurements, you get between 1,000 or even more proteins, and metabolism the same. And so that's where the huge amount of data is coming into play. And then, of course, we have very different data types. So we have large number of features and very different data types. And that's where the need for data science then arises. Okay, and we are for sure not at the end of all the development. So this is very rapidly evolving technology. So it's like we are really just started to open our eyes, and we are seeing more and more what's happening. So that's the half of the product panel of one of the leading companies here in the sequencing technology. And you cannot read that, but just to illustrate that. So each line here is a graphical summary of one of their technologies, which they offer. And each of these technologies give different aspects of the molecular organization of a cell. And this is really evolving quite rapidly. So we are seeing more and more the different details. But the question is now, how should we use all of this information? That's absolutely not clear. And people usually use a part of it, so they do this top 10, top 100 differentially expressed genes or things like this, and then they look in the list and stop where they find then their favorite gene, and then they look into that one. So that's a completely 99% unused information, I would say. So I think I skipped one slide here. I should go back to that one. This one. So this is just it's a nice illustration on how the omics took over publication in life sciences. So this is starting from the year 2000 till now. And you see the top one, this is really genomics. This was around for quite a while already, and there's a lot of development already in that area before 2000. But now it's on a constant production level, I would say. So this is log scale here, as you can see. And then other technologies were developed in the early 2000s and then also reached very productive levels here on. You see proteomics and transcriptomics was really speeding up a lot, so there was a lot of development here. And there is also this, maybe you can see that, this multi-omics approaches where people try to use several of these omics. So it has a very, large growth rate here, but it's still in low percentages compared to the other omics studies. I'll get it till the end of the talk, I hope. This is the amount of data, also for the different data types, which is stored in the EMBL EBI data resources. So this is around 2 years ago, 160 petabytes of data. And as I said, I think most of that data is really not used. So there is on the surveys, we pick the favorite things, but I think most of the data is just not used because there's no, I would say, not very many efficient methods to really dig deep and harvest all of that. So I'm pretty sure that there are many more things hidden, like the CRISPR-Cas technology, which just was awarded the Nobel Prize in Chemistry. I think there are many such things hidden in this data. It's just a question how to find them. Okay, and then of course, if you think about data, we are not just having this omics data, genotype, transcriptome, or methylation data here on that example, but of course there's also some metadata always coming. So this is from the Cancer Genome Atlas. If we are in my field, so if there's a cancer patient in the hospital, then usually the medical doctors take a biopsy, so take a piece of the tumor out, and then this is analyzed. And then you can do the genotype, so that's what's usually done, that you get one or few mutations of the cancer, and that helps already in the decision of the treatment. But now more and more, the hospitals are also using these omics approaches to deeper analyze the cancers. And of course, there's a lot of metadata coming with such a sample. So about the patient, so that is demographics data, age, where is it from, then which treatments were given to the patient, did it work, and so on. So there are many additional data which also need to be handled in a proper way. Overall, there are quite some issues related to data science in our field. So that's a nice summary from one publication. There are things, just starting at the lower left here, more on experimental work, so sample preparation. How do you extract in a really solid and robust way, for example, RNA from such a biopsy? How is this done efficiently? which study design to use, etc. So you can read a bit statistical power calculations. This is still also a very important issue and not always considered sufficiently. And then there are more other things like each of these technologies needs an own normalization and data transformation approach. Usually there is a statistical model behind it. There is missing data which need to be imputed, and so on. And then more data usage topics like workflow and pipelines. So is it reproducible what you're doing, and how is the archiving done and all of that? So that's then moving into very general data science topics, which also other fields will have for sure. And then the big question on really how to integrate this data, so not just look at one data type at a time with a statistical model, but how would you combine things, and how would you then extract really knowledge from this big pile of data. Okay. So this was what I wanted to say just in terms of introducing the data science in life sciences. And I would like now to move into a direction of precision medicine, which is the area we are working ourselves. But of course, all these omics approaches are used in many different fields in life sciences. So this precision medicine is not just our topic here at the department in life sciences and medicine, I would say. And what in the end we would like to do is to be able to tailor the treatment to patients, so not have one treatment for certain cancer type, because we know this is quite inefficient, but to have more tailored and by doing that more efficient treatment. And the question, can we use all this additional data which we are generating to really tailor treatment and get precision medicine? So can we, and this usually it's trying to be done by either single biomarkers, so that's what I said. Very often this is mutations, key mutations in driver pathways. And now since quite some years there's also the idea of using signatures, so a panel of such markers used together. And the hope is then to increase the predictive power by combining these markers. And then the question is, okay, can you even go beyond the signatures, so use more this type of network information? So where you would not treat the markers as like independent entities, but because that's not true. These markers are never independent. There is always a kind of link and dependency, a network link. So how can we make better use of that type of information? And there are two major problems from my point of view, which I would like to introduce here. And I would like to give credit here to Harald Schmidt from Maastricht, who was so kind to borrow some of the slides I'm using now in the next few minutes. One of the biggest problems is that existing drugs really have a low precision. And this can, for example, be seen in these numbers needed to treat. So that's an important measure which can be considered when treating patients with a certain drug. And we see these different groups of people, of patients. And these are the top 10 drugs which were sold already a while ago, 2012, but it didn't change a lot since then. And you see that only the green portion of patients is responding to a treatment. And all the red ones are not responding. And this is not like an exception, and these are not the bad cases. So this is the average, what you are having in treating patients. So many patients are just not responding to the treatment. So this is a lack of efficacy in the treatment. And that's, of course, for cancer patients, for example, if you stay with that topic, that's a very bad situation because they usually don't have time forever. So there you have maybe 1 or 2 chances of treating such a patient, and then it's too late. So it's really very important to find the right treatment right from the beginning. Okay. And this is also true if you look into clinical trials of new drug development. So this lack of efficacy is also the major reason for drugs to fail in the clinical trials. So they look promising in the preclinical studies, and then if you move later to the clinical trials, it just doesn't work for enough people in a sufficient way. And that could also of course, be a question of the selection of the people you have into such clinical trials. So maybe the drug is working, but not for all of the people you would like to treat. So the question is the selection also of the people you have into these clinical trials. The second big topic is the clinical safety here, which I will also come back then in a minute. Okay, and a lot of a discussion in that field is going on the question, are our definitions of the diseases we are using, are they still up to date? Or are these categories too broad? So there are, for example, like Parkinson's disease. This is named after a doctor, and the person or the doctor observed certain disease with certain phenotypes, and then it was named like that, and everything was grouped under that umbrella. But that's maybe not the best classification. So very often there are subtypes, and especially also in cancer, there are subtypes of different cancers, and not all of them, if you call that a colon cancer or a melanoma or whatsoever, it will not be the same disease in the end, even if some of the phenotypes are shared. So the question is, can we now be a bit more accurate in our disease definitions, and by that maybe also more selective in the drugs we are giving? So the field is moving really towards this network medicine, trying to use the omics information also really as a additional very important piece of information to tailor the treatments. And this, of course, not readable, the slide, but it's just to show you that if you go to this list of different diseases, and you see that, for example, the cancer, which is a very big cluster there, in this light blue color, it's of course, there's a lot of connection between the cancer types, but there are different subtypes clearly visible. And there is also a strong neighborhood, for example, to the neurodegenerative diseases. So there is some common mechanisms and common molecular features which are shared between neurodegenerative diseases and cancers, also between cancers and metabolic diseases. There is a lot of shared molecular features on that. And that's also resulting in so-called comorbidities. So people are having two or more diseases, and so there is that's what usually happens. So there is a mechanism below, something is going wrong, and then there are some phenotypes on the surface which might not seem to be correlated, but if you really look deep, they are correlated. There's some common root to these diseases. So that's where the field is moving to. And the second big problem which we have here in the drug development is that the costs are really exploding in drug development. And this is a plot basically on the number of new drugs approved per billion US dollar over the years. And we see this is also on the log scale, it's a pretty stable trend here, so it's becoming more and more expensive, and people call this also the Irum's law, so the Moore's law inverted. It's getting more and more difficult to develop new drugs and to have them approved on the market. And there is a lot of discussion about this will not be sustainable in the long run. So this is getting to the end of the pharmaceutical industry as we know it, because it will just not pay off anymore to do these drug developments. And there is also now, based on that problem, there was a new development, and I would like to show it to you if I manage. Okay. And that's related to the second link. This is the second point on the failure in the clinical trials, which is the clinical safety. So the drugs look safe in the preclinical studies, in the animal models, in the first cohort of people. But then if you move to a larger cohort of people, then there is some side effects and some really severe side effects popping up, and it's not sustainable anymore and not possible anymore to move on. So clinical safety as the second reason. People are trying to overcome that by doing something called repositioning of approved drugs. So basically relying on drugs which were already approved by the agencies for one disease. And based on these common joint mechanisms in the different diseases, trying to see if we can use such a drug also in the context of another disease. So this is repositioning, move it from one disease to another disease. And for example, I will then show you in the examples that we can do that also for cancer treatment based on our network approaches. But that's really a growing field based on the fact that already a lot of money was invested in the drug development, and very often it didn't pay off because the drugs didn't reach the market or they were not effective enough for enough people. And very sorry here for the... Okay. So this is just a screenshot from a table from a literature review on drug repositioning. This is not something which is very new, so people are doing that since a long time. And what I really find interesting here, we just need to go to the end of the table. The first column are diseases per drug. So this is how many diseases are treated with one specific drug. And you see there are many drugs which have up to 300 and more diseases. So you can use one drug and treat very different things. And this is not very similar diseases. Very often this looks completely different from if you just look from the phenotypic level without digging deeper. So there are 189 drugs which are used for treating more than 300 diseases. And one interesting example is the chloroquine. So this drug was in the discussion now a lot with COVID treatment. Maybe you followed that a bit. And it's mainly used for treating malaria. But you see from that chart, since it was developed in 1984, there are more than 400 diseases now considered to be treated with that drug. And every year there is a new disease where people are considering that. And the red line is then where it's really moving into clinical trials. So this is of course less. This is a bit above 50 diseases. But so chloroquine is one of these drugs where you say, okay, let's try that. And then sometimes it works, sometimes not. And this is of course a very random trial and error approach. And we should try to become smarter in picking these drugs for repositioning based on all the data we are generating. Okay, I don't want to move too deep into drug repositioning because this is really a topic on its own. There could be a separate lecture on that. There are very different approaches, a lot of computational approaches also trying to work on that. In the end, you could look into drug-oriented, target-oriented, or disease-oriented approaches. So it basically says if you have a drug, you look for a new disease. This is what we often tell to pharma industry they should not do. Should not come up with a new disease and just to sell your drug. But sometimes it's not that easy, so it really makes sense to do that. Or you have a certain disease and you would like to find a new drug for it. And I will show you a bit of our work then in a minute. And yeah, as I said, there are many different computational approaches. People are, for example, also using literature information or Twitter messages to try to do this kind of repositioning. Okay, and last slide basically before I move into our work is these so-called comorbidities. I talked about that, so diseases which are showing up from in one patient, similar diseases or linked diseases, and I hope you can read that. So it's basically lifestyle factors here in the columns, and the association to cancer types in the rows. And these darker ones are the ones where there's really strong evidence for negative impact of lifestyle factor on cancer development. And this is body fatness, alcohol, and smoking. And that's really a very strong evidence that this has negative impact. So smoke, alcohol, and fat. And that's something we can also try to use in terms of the fatness and the metabolism, and explore the link between cancer and the metabolic diseases. From these studies, it's very clear that, let's say, the prevention of cancer is much more promising than the treatment later. So it's not so easy to say, but in the studies where people try to estimate that, usually they say it's around 1/3 of the cancers could be prevented by really looking into these lifestyle factors. And 1/3 is really a lot. And another 1/3 could be treated easily if we would just have early detection. So that's the second big topic. And the last third is then when it's too late. So when the cancer really starts to develop, and we need to find a treatment for a system which already tipped over the tipping point. Then that's the more challenging part. And that's also, of course, where we are working in. But just to remind everyone that lifestyle factor and early detection is really super important for cancer prevention. Okay, so that was the long general introduction on data science and where we are aiming to with this drug repositioning. And in the second half of my talk, I would like to show you 2 examples where we are trying to contribute here to the field. And the first one would be from this problem area 1, that existing drugs lack precision. And we are developing also, of course, in strong competition with other groups, also a drug sensitivity prediction pipeline using machine learning approaches. So we try to predict from these omics data which drug would be effective for which cancer. So should we treat a certain patient with a drug, or should we better use another drug? And machine learning can obviously help quite a bit on that. And we are usually developing the pipeline and the basic models based on cell line data. So these are cells which are taken out of the body context, and they're grown in the lab. And then, of course, they are changing already a bit by doing that. And we are still using these cells as models for the real cancer, and knowing that this is not true, of course, but yeah, there's still some truth to that. And on these cell line panels, like here, 450 cells, we can then more easily do all these measurements, which are needed to train machine learning models. And the data here are used from Cancer Cell Line Encyclopedia, and that's a variety of cancers, as you can see in this pie chart, without reading, but that's just all different cancer types, like lung cancer being the most represented there. And then we see on the right-hand side then the different data sets which are reproduced. So we have these drug screens where basically tested in all of these cancer cell lines a variety of drugs and checked if they are responding or not. And then we have RPPA, which is protein measurements, and RNA, so transcriptomics, and microRNA. This is also a different layer. And this was not done for all of the cell lines, so we used these 451 cell lines where all of the data are available and moved then into the machine learning pipeline. And there is always a bit of data pre-processing necessary before we really can, of course, feed the data into machine learning. And in this case, for example, we need to think about how can we use the cancer drug treatment data in the machine learning pipeline. And what you can see on this graph is basically on the x-axis the inhibitor concentration of the drug, so the drug concentration which is given to the cells. And on the y-axis it's the growth. So is the cell still growing or not? So that's what we would like to prevent in cancer. Cancer should stop grow, and ideally we should even kill, but at least stop growing. And we see that if we increase in this situation, if we increase the inhibitor, then at one moment it will work and the growth is decreased almost to zero. So this is like a nice example where the drug is working at a certain range. And what we usually use in these models then is the so-called activity area. So this is this area above the inhibition curve. And that goes a bit beyond what's often done, namely using the IC50 value, which is the point where you have half of the growth only left. Because sometimes the curves is not that nice, and then the activity area gives you just a better representation of the drug's efficacy. And then in the second step, we would then get such distributions of these normalized activity areas, and we have these three colors. So this is one distribution of the 451 cell lines. And the question, are they responsive to a certain drug or not? And then there will be like the green ones. We consider them to be responsive. And you see that there's some which are really completely responsive and other just a bit. And then there are the red ones, which we consider not to be responsive, so the so-called resistant ones. And also the question on how would we discretize the data on that stage, this is also not really fully solved. There's a lot of manual tuning and fiddling around and looking at distributions. And for each drug, these distributions, of course, look a bit different, so there is no easy standard way to do that. So that's what we use in the moment. We just call the upper 1/3 sensitive and the lower 1/3 resistant. But this might, of course, not be accurate given a certain distribution here. Okay, but that's what we use in the moment, and then we are moving into a machine learning pipeline. I didn't bring too many details here, but I think that's basically, we are people applying machine learning, so there is not a lot of development on our side. So we use the existing toolboxes and try to feed all these features. We do a bit of feature engineering in terms of using also pathway level information or some kind of network information. And that's also active research, so there's for sure more to be done, or information on gene ontologies, so sets of genes which are grouped together based on their functionality. And in the end, we can then, that's in collaboration with a small company here, started by a former postdoc. So they are using their model stacking to get pretty nice accuracies for a number of important drugs, so above 90%. And that's something which is already beyond the state of the art, I would say, in the field. And I think a lot of the success is really in the data pre-processing, in the cleaning, and making sure that you feed good data into the pipelines. And then, of course, the question is also, can we understand why we get a certain predictivity? So what are the key markers we should in the end measure in the clinical lab, so that we can, for a specific patient, tell if we should give the drug or not. So we need these markers or these marker panels, these signatures, to be able to move into application in the long run. So So this is just one of the data sets. As I said, this is really work in progress, so that's not published yet, and it's also not completely done, but just give you a feeling what we are looking at. So this is for each of these panels there is one drug. And for each of the drugs, basically, we built these machine learning models and we tested which of the underlying data set is really important to achieve a good predictivity. So would it really make sense to measure all these proteins or the mRNA level? Is this really relevant or not? And then we also did some tests with principal components here. This, as I said, this is a rather simple approach, but the tendency we see is that the light green bars, which are the mRNA levels, so the transcriptomics seem to have a lot of information already, and it's not so far from the complete data set, which is the dark green. So it looks like we can really make use of transcriptomics only to have already quite predictive models. And that's, of course, if you think about application in the long run, this is the good news because you don't need to measure all of these different omics types. It's then sufficient to start with transcriptomics, which is also getting more and more cheaper and doable also in the lab. So that's something where we are currently moving in also with partners and see if we can really do that also for patient data. Because as I said, this is mainly cell line data, and yeah, that's not the ground truth here of the cancer. Okay, and this is just, it's not supposed to be read. This is a poster by a PhD student, joint PhD student with the LIH. We also tried just to advertise a bit that UA here is working also on interpretable machine learning models, namely random forests, where we try to really extract information why certain decisions are taken. And there is some interest also from him to talk with people working on similar work. So if there's anyone, then let me know. Okay, so with that, I would like to finish this first example, if I manage, and use the remaining time to look a bit more detail into the second project I'd like to show, and this is on this problem too, namely the fact that new drug development is very costly and very often unsafe, so it fails in these clinical trials. And can we use drug repositioning of already approved drugs to avoid these safety issues? And here we are developing a network approach since many years. And the idea is basically to really make use of this network information. So as I said, not to treat the measurements of mRNA as independent individual features, but you really use the network dependency they have to get better predictions. And there are quite nice existing methods already for network analysis. So coming from engineering also, or within the systems biology community. So if you have such a metabolic network here, then you can quite easily figure out what are the sensitive nodes. So where would you need to target the network so that it fails if you want to target a cancer cell? And especially, for example, if you have such a metabolic network for cancer and one for all the healthy tissues in the body, is there any position in the network which could be targeted so that you only have a strong effect on the cancer but not in all the healthy tissues? So we would like to have these selective drugs. And for doing this, the methods, I think, are quite robust and good. The problem is really the construction of the underlying networks. So if the networks are not of good quality, it will not really work. And that's why we spent quite some time on the data integration and the reconstruction of such networks. And over the years, we developed, so that's now really the core activity, I would say, of my group, is this data integration and network reconstruction. So trying to have really best models you can get with the data available, by using as many of the data sets which are given for a certain disease. So that's what I consider data integration. Use the diverse data types and feed it into one computational model of a system, of a network. And as I said, yeah, we worked a lot on metabolic networks, and that's what I would like to show in the remaining minutes. But we have also worked on signaling networks, which need a bit different treatment because of some details, not to discuss now, but there are some specifics there. And also gene regulatory networks is also something which is ongoing, also in collaboration now with computer science, with Yun Pang, trying to advance the field here. But yeah, today I would like to talk about FastCore, so which our main working algorithm for the metabolic networks. And such a metabolic network, just to say a few words, that's what's depicted there in the center. It's basically comprised of metabolites, which are the nodes, and reactions, which are the edges. So metabolites are converted with reactions into other metabolites. And by doing that, you get this huge network. In the end, it's basically we take food into our system, and the metabolic network of a cell produces whatever is needed for growth or for fulfilling its functions. It's not always growth. It could also be other metabolic functions. And yeah, as we know, for cancer, the growth is out of control. And so we need to try to understand what's happening there and can we selectively target these cancer networks. Okay, yeah, this is just, maybe not talk about that. I mentioned that already a bit. Yeah, then all of them come at once. Okay, so what we basically want to do in that project area is to identify cancer-specific metabolism, so what's really specific, and then exploit these vulnerabilities which show up and find specific drugs. And maybe already to summarize the key finding we have there is that cancer metabolism compared to healthy metabolism is usually more compact, so less pathways used. It's a bit like a Ferrari, so it's streamlined, and it's not so robust anymore because you have all these extra backup pathways and safety mechanisms which a healthy cell has are usually kicked out by the cancer cells to enable faster growth. So they are not producing all these extra enzymes which would be needed, but stick basically to the core machinery. And by that, this core machinery is becoming more vulnerable, more sensitive, because these extra backups are missing. And that's what we try to find. So where is these new sensitivities in the cancer metabolism, which were not there in the healthy? Okay, and how do we do that? Yeah, so there's a lot known, of course, about mutations in cancer, but that's basically, I gave you already the summary of this metabolic rewiring. And the workflow here is more or less as I showed you already. So we are, in this first version, we are using expression data, so mRNA levels. That's what we saw from the machine learning studies is most effective for really giving the drug sensitivity. And we are combining the expression data with some generic reconstruction. So this is basically like a database download of all the metabolic reactions which were found in humans in very different cell types over the years in different contexts, and not all of them active at the same time. So this is a excuse me, it's a pretty wild collection of knowledge. And then we combine these two data information into one computational model using the FastCore algorithm, so this is now really the topic of contextualization. So we make this generic model context specific for a certain sample. So this could be patient specific or patient group specific. And then this context specific model is only a subset of the metabolic capacities of the full model. It's really what's active in that Ferrari now, and not from everything is there. And this is, of course, might even be a bit readable. But as I said, I will not look so deep into details, but we are using here a linear programming-based approach. So this is very efficient, and it allows us to handle networks of thousands of reactions in a quite efficient manner. So this is a two-step LP approach. The first step is pushing basically these red reactions, our so-called core reactions, into the model, because we know they should be there. There is some evidence from the expression data, so we want to have them. And the second step, the blue step, then tries to minimize all other reactions. And this would work if they are not necessary to have a flux in the red reactions. So, yeah, that's the basic idea. I think I will not dig deeper here, but of course we can of course talk a bit more. And this algorithm we tested that against other methods, of course. So there's always this benchmarking efforts in our field going on to indicate what is what is working and what not. And I think we had quite good benchmarking results for our methods, but there's also things which are still not working, so we are also developing that algorithm further. But we are fast, pretty accurate, and have a high resolution. And I think the basic philosophy of the algorithm is to have pretty compact reconstructions. So we try to stick to the core evidence. And yeah, I'm not sure if I should talk too much now about the details of it. It's getting already a bit long. But also here, just in terms of the data science, there's of course also some pre-processing necessary. And also here on the RNA-seq data, there is a discretization step necessary. So we need to basically tell then later for the algorithm which of the reactions we consider as core. So where is sufficient evidence that we would like to have them in the model? That's what we say is a core reaction. And there is not this. We never get this discrete information from our measurements. So there is some kind of decision making here necessary. And yeah, as I said, I don't think I will look too deep into how that is done. But that's basically some statistical approach to do that. Okay, and also we did, as I said, we did this benchmarking. And I'd like to just talk about that here a bit. So the big figure here is database information, basically showing for a given reaction in how many tissues would you find it in the body. So if you are interested in a certain reaction, is it in all of your tissues or just in specific ones? And what you usually get is this kind of distribution that there is only very few reactions, so the long tail, which are present in all the cells of the body. Usually you will have the metabolism of one specific cell will be specific. And that's what you see in this kind of distribution. So the most of the reactions are only present in very few tissues. So there's a lot of specificity in our body. And if we compare that, on the other hand, with the model reconstructions, then we see that most of the algorithms are not giving us such a specific distribution here. Just the high resolution algorithms which we produced did do that, so we have this typical shape which we should get for all the reconstructions. Very often in the other algorithms you see basically a lot of the reactions being present in all of the cells. And that's of course not specific, and this will not give you specific predictions later. Okay, but maybe, as I said, we'll speed up a bit and just show you also the results in the end. if I manage to move it. So yeah, that's something we of course should still mention, that we are also contributing to community efforts, and I think this is a nice example where the community of these constraint-based models is really working together since many years. And that's now version 3 of the so-called Cobra Toolbox, was published last year. And yeah, it's not so easy to maintain these huge software projects in academics. So very often they are then disappearing after a while, but that's one example where it really works now since 10, 15 years, maybe even longer. I'm not sure when exactly it started. Okay, so we did that, this reconstruction also once again for a cell line panel, 10,000, this is patient data, for a patient collection of 10,000 patients. So we took these samples, reconstructed all the models, and then specifically looked into colon cancer and tried to elaborate now which, I'm really sorry for that. So. We look basically into rat cancer models versus the healthy model for colon, but also for all the models, and we see that usually the cancer models have less reactions. So the red bar is a bit lower. There are some reactions which are kicked out in the different pathways of the model, and that's what we, as I said, what we try to make use of. And the application is now this drug prediction. So the full pipeline would look like this. We do the reconstruction of the models based on this expression data in a generic model. We get this context-specific model, and there we can then do knockout analysis. So we remove one gene at a time, computationally, and see if it would have an effect or not on the growth of the cell. And as you now have a computational model, this can be done very efficiently, and you can get such lists of candidate genes which look promising for targeting the cancer, but would not harm the healthy controls a lot. And from these candidate genes, we picked some of them and validated them in the lab in collaboration with a group here in the department, so the group of Serge Hahn and Elisabeth Letelier. We validated some of the predictions for colon. And just to show you the results, moving a bit faster here, so there's a number of drugs we could find. Some of them were already known cancer drugs, so that's good that you can find them back, but there were these 17 which were so far not approved for cancer. So this would be good drugs for considered for repositioning, because they were used for inflammation or different things, but not for cancer treatment. And we tested 3 of these non-approved cancer drugs, and could find now that the upper panel here, I'm sorry. So the upper panel, these are our control cells. These are colonic fibroblasts. So this is from the tissue around the cancer. And there we see that for the 3 drugs in the columns, if we increase the concentrations, there is not a lot of response. So the growth rate is not really slowing down. This is the growth of a cell, so it's just continued to grow. While the colon cancer cell line, which is coming from a patient, from a hospital here, it shows that with the same drug concentration, the growth is really slowing down a lot. And that's the evidence basically that for at least for this small experiment we did here, that we can find something which is specific for a patient and not harming now in these fibroblast cells which are in the surrounding of the cancer. And that's, of course, only the first step. So this is just a very small example. We are now moving on in the experiments to also look for combinations of drugs and having more control cells, also looking into other cancer types like melanoma or head and neck cancer. So we are working there with other groups to just get more evidence that this type of network-based repositioning is really working. Yeah, and with that, I'm I think I'll stop here. There are a few more slides, but I will not show them. So the idea is, a summary, so we can identify cancer-specific metabolism using these network approaches. And I didn't talk about all of that, but what I explained is that these cancer models usually are more compact and thus more sensitive, and we can really exploit these new sensitivities and use then other combination with other database knowledge to identify already approved drugs from other diseases and use them as candidates for this repositioning or repurposing. Okay, with that, of course there is outlook bit. So the next step, as I said, we move to other cancer types and try to see if things are also working. We look into combinations, and this is now the subgroups. So we try to zoom into the colon cancer subtypes which are available. So not all colon cancer is the same cancer. There are these different versions, at least 4 different types of colon cancer. And we see if we can also get some more specific predictions here. Okay, with that, I just would like to show you the team at the end. I can find that. I skip over the remaining slides. Okay, I was expecting that this was happen. Okay. Yeah, thanks to the team. Some of them are here. It was a big effort from many, and thanks for your attention.
So thank you very much, Thomas, for this very nice talk. I've learned a lot about research.
That's nice.
So as I said, this workshop is also based on interaction with the audience. So please, do you have questions to ask to Thomas? Go ahead. Yeah. Oh, wait a minute. So there will be a mic coming to you. So in fact, when you're seated, you can take off the mask, but when you ask a question, please put it on. So
I was wondering, we're focusing on big data, right, in principle, in this workshop. But if you think about animal models and drug development, I think that's actually really small sample size, right? Am I correct about that?
The sample size usually is a problem, yeah, exactly.
I was a bit shocked when I saw that the drug really worked. You show it with these like red and green images of like people. How do you say that? That it's not so effective already so far actually, and now it seems that they try to reduce the cost and reinvent the drug already now in the market. That's how I see it after your story. Mm-hmm. Would there be any way to maybe also improve that part of drug development?
I'm not sure if I fully get, so the low sample size basically?
Yeah, because maybe they will, I think there's always a lot of pressure in drug development to find some result, and I'm not so sure if it's always really neutral, to be honest. So that maybe sounds a bit negative, but I don't mean it bad that way. But maybe sometimes some effect is also kind of created almost. If you also have your distribution where you had training data, 1/3 of it basically out, that doesn't feel so good to me as an econometrician, that you throw away information, and also maybe it creates some effect, but I don't know anymore what I'm really looking at that time.
I think you're right, yeah. That could be really a bias which is introduced there.
Because also with the machine learning techniques, you need to be careful a little bit what you're in the end looking at, because like supervised or unsupervised learning, but if you create your own binary variables, for example, 0 and 1, and you kind of fabricate it and then do another analysis and so on, then maybe you don't really know what you're looking at anymore. So, but it's really interesting to find some patterns. It's super interesting.
I think that's completely right. We are. That's why I have a lot of interest to be here and discuss with you and show what our problems are. So then most likely there are very smart approaches already available, and we could just use them. But
And
we need
also
to figure
maybe a
out that they
small
are
detail,
existing.
Maybe about measurement error or something. That's also typically what we encounter. I may assume that's also maybe something you will encounter.
No, for sure. There's a lot of didn't talk about that. There's a lot of measurement error always on different things. It might be very different for the different techniques. Some are pretty precise, others not.
Okay, and then one last question came to mind is, do you think that the currently available drug, there's still a lot of opportunity to find new purpose for it?
Absolutely, yeah. I'm completely convinced.
The good news, right? That we can still try to improve the treatments.
Yes.
Yeah.
I'm not an expert, but my feeling is that most of the drugs are underused. You know, they are. Of course, it's always a marketing issue also for the companies. They have their portfolio and they need to find the correct market segment to push it. But there are probably many drugs which could be used for smaller groups and more precise. But to figure that out, I think that's the challenge. And then, of course, to convince the companies to go into that direction, that's then another step.
Thank you.
Thank you.
Other questions? Yeah.
Yes, I have a couple of questions. First, you mentioned the availability of data measured in hundreds of petabytes. So is it publicly available or subscription-based? Is there a paywall?
No, this is the EBI, so the European, what's the name? Publicly funded laboratory. So this is available. So most of that data you could get. And of course, there is much more data also in companies, which is not publicly available. But there are some initiatives where companies put out their data or part of it and basically have these, these challenges, data challenges, so that try to find someone who can do something useful with it. That's also happening, but most of that data I showed is publicly available.
That's great, but another thing for replicability, you need not only the data, but the code. And on one of your slides, I think I've seen the Excel icon and MATLAB icon and the toolbox, which is essentially a black box. And if you remember recently, I think last week, the UK lost 16,000 records in COVID cases just because the Excel file ran out of size. So are those algorithms that are being used in that research publicly available so that everyone could get the same reliable results and really verify what is going into the black box?
Sure. Yeah. Notice these algorithms I presented are publicly available. We are still doing, for the network approaches, quite some developments in MATLAB, which is, of course, a commercial package, so you would have the license for that, but a lot then, once it's working, a lot of it is then also transcribed in other languages, in Python or R, which are then completely free, and the algorithms are available.
Oh, that's really great. Yeah, thank you.
Sure.
That's very sure.
Yeah, there's a question here.
Should I hold it?
Thanks, Thomas, for this wonderful talk. I have a bit of more of a Gedanken question here. So most of the data you showed that pertains to cancer research, but there is a growing evidence that cancer and microbiome, which itself is a big data pool, they kind of cohabit and co-regulate response and emergent therapeutic approaches. So do you think there is a need to kind of build up innovative models that could combine these two big data sets that we generally associate with either cancer research or microbiome research? And if so, what could be a good way forward?
I would agree, as I'm not an expert in the microbiome, but from what I saw in the papers, I think there is enough evidence that there is some correlation or even mechanistic link. So it would make sense to explore that for sure. And there is of course these already efforts in terms of generating not only one cell model, but these combined models and higher level tissue models. So I think that that's ongoing. Of course, it has new technical challenges, how to do that properly and make sure that the models are still predictive. But yeah, I think that's a very important direction to follow up. I skipped over one last example where we did also some higher level modeling, combining these specific reconstructions for key tissues in the human, so fat, muscle, and liver we used. With these three tissues, we could then quite nicely model the metabolite concentrations of certain inborn errors of metabolism. So I think that's a very promising avenue to follow.
Thank you.
There are questions over there. Oh, okay.
So Anupam just mentioned another omics and said, can you link them together? But there's not just two omicses. We have environmental stuff, I think, for the next talk. So
Sure.
Can I ask, is omics a buzzword? Do people do omics?
Omics, yeah, probably. Sure. No, sorry for skipping over many of these omics. I'm a bit biased there in the data types we are using, but yeah, you will see more in the next talk for sure. I think maybe just picking up on that, so omics, there is a bit of, I'm not sure what's the English word for that, but Goldgräberstimmung, so you're just harvesting the data and you produce the data and get as much as you can. And I think that's fascinating because we have these new technologies, but this data is just stuck and we need to do also the processing step and also have much more developments there, not just produce the data. Not sure if this is answering your question, but that what comes to mind.
Sure. Well, I mean, I'm sure hundreds of people have grand frameworks which can propose to unite all of this stuff and to make an inference based not only on,
Sure.
whether the sun is shining today and what you had for lunch and also your genotype.
Yeah.
I guess somebody is doing that.
No, absolutely. Absolutely. People are working a lot on that. That's what I showed you with this curve on the multi-omics publication. So this is really speeding up.
All right. Okay, multi-omics, that's what it's called.
Yeah,
okay.
the multi-omics stuff. So this is really speeding up, and I think that's absolutely necessary to have that. It's just not in the daily routine of a lab. This is not happening so often.
The omniomics also sounds good. Omniomics? But,
Yeah.
okay.
Thank you.
Other questions over there?
Yeah, so first of all, thank you for the talk. And I'm starting working with networks analysis, and I was curious about what you mentioned about networks. You explored where the nodes were the metabolites, and the edges were the reactions. And I was wondering if you also explored other kinds of networks, for example, correlation networks, where the edges would be associations or correlations between those metabolites, how they are expressed.
We didn't do that in the context of metabolic network modeling because I think that the evidence you have from the biochemistry in the last 50 years is quite useful on what edges are existing. There might still be missing reactions which are not found yet, and then maybe such correlation studies might be helpful, but that's usually not done in the context of metabolic networks. But it's done a lot for signaling networks or gene regulatory networks, where it's not so easy to have the certainty about the edges. So there's a lot of mechanistic studies there needed still to clarify which edges are existing, and then people use these type of correlation approaches to reconstruct the networks.
Okay.
Yeah.
Thank you.
Sorry. Thank you for the talk. I have a question about the prediction that you were introducing. indeed you have been talking mainly about prediction, but I was wondering if you want to say something that is working or not, you should switch to a kind of causality approach. So, for example, in the graph that you showed before, when we look at the graph of a given drug given to a given patient, okay, we see that the drug is working for this person, but we don't have the counterfactual situation, like a similar person or the same person treated with a placebo solution, or in general they also use an alternative treatment. So the gap between the placebo situation against the contrasted with the treated one should be considered as really the effectiveness of the treatment. Then
Sure.
I should absolutely probably consider different long-term versus short-term evidence, and also how in a kind of dynamic sequence, because I think that also what is happening now in the short term is also influencing the long-term effects. So I don't know whether you are considering to introduce a kind of dynamic approach and a causality approach as well. That is a key, let's say, factor in this framework of treatment evaluation.
Yeah.
Thank you.
Thank you. Yeah, there's not too much to answer. I would fully agree with what you said. I think that's necessary. What we did so far is, for me, a bit like a pre-screening. So we tried to come up with a couple of candidates for further, more detailed studies as you outlined it now. So we, of course, for all of these drugs, as they are FDA approved, there is some mechanistic evidence what they are doing and what are the targets. So this is known, but of course, if you go into a new disease, then there would be some more mechanistic validations necessary if you really want to push it into the clinics. So that would never be done by just such a model alone. And also of course, the long-term effects, etc. So that's something which is important. Yeah. The good point is that these drugs are already in use. So they are already treated, patients are treated, and it's safe to do it. So, but of course, it could, the long-term efficacy, this is also something which is not trivial to model. I wouldn't know how to do that in the moment. But you're absolutely right, so there is more work needed there.
We have time for one question here, I think.
Thank you very much for the nice talk. I have a question about proteins expression. So, in your modeling, are you taking into account the specific expression of protein of a human being at a certain moment of his or her life? Because, you know, proteomics is the full set of proteins we can express inside us. But at a certain moment, we express just a smaller set of this full set. Mm-hmm. So maybe if you consider a patient that is treated for several years, this patient can also express at different times different type of proteins that react differently to the drugs that is receiving. So my question is, do you consider this change in the proteins expressions in a patient?
I don't think we are doing that in the moment. Technically, it would be doable if we have the information. We could consider it. But I don't think we are running something like that in the moment. But yeah, once again, you are right. So should be done.
Yes, thank you.
We have, we are operating, maybe just one comment here, we are operating on steady state assumption for these metabolic networks. This helps a lot to simplify the modeling and the analysis. This is nice, but of course the steady state assumption is not true. So in the end, if we would be in steady state, we would be dead, and that's not the case. So there is always something happening, and that long run adjustments are very important, of course. But in the moment we are not looking into that.
Maybe last question. There was. Okay, here.
If I may add a question to my previous one, indeed you introduced, I think, quite fast, but of course there are plenty of underlying assumptions in your models. You mentioned, for example, missingness issues, missing data issues. I think that also the way you input the missing data is extremely important, and probably might be also relevant to see, to make some, because you also introduce sensitivity checks. I don't know which kind of sensitivity, but let's say just compare and contrast, like via simulation studies, how much robust are your results compared to the underlying assumption. If you change, for example, I don't know, the missing at random assumption for missingness to not at random assumption, how much the results that you have might change, and also how much your machine learning tool might change in the selection and prediction that is implemented in your studies, for example.
Yeah, once again, I think you are completely right. So we are, this is an important issue. So all the modeling results are, of course, depending on some of these assumptions, how you do the gap filling. For example, in the modelings, we have in these metabolic networks always these gaps where just not enough biochemical evidence is available. Then we need to do gap filling, and there are different ways of doing that. Or you ignore the things completely, but then it's the risk that you kick out important pathways. And this is, of course, all of that is influencing the results. So I think in biology, we unfortunately are really in the situation that we have a lot of uncertainty we need to deal with. It's like just coming back to this early example of the Lego box. You know, you have these 2,000 parts there on Christmas Eve, and then you take some of them and build something, but it will always only be a subset if you don't have the complete plan, and this will influence the outcome quite a bit. So we try to find our way, but there is so much uncertainty we need to deal with.
Thank you.
Okay, thank you very much. I think it's time we move to the next talk. Thank you very much again, Thomas, for this.
Thank you.
Beautiful.